Hello, this is Pallavi here again with another video, and in this video we will be talking about mitochondria. Mitochondria is an important cell organelle which undergoes very crucial processes like oxidative phosphorylation and which is also called the powerhouse of the cell. In this video, we will study how the mitochondria was originated, its ultrastructure, and also its basic functions. So, without wasting time, let's begin. So, let's begin with the discovery of mitochondria. It was discovered by Colliker in 1880 in muscles of insects, and the term mitochondria was given by Benda. The mitochondria are actually self-duplicating organelles. That is, they divide and they form two cells, similar like binary fission. And the size of the mitochondria varies from 1 to 4.1 micrometer in length, and its diameter varies from 0.2 to 1 micrometer. So the evolution of mitochondria is very interesting. Previously, it is thought of as they were independently living organisms, and as time passed, the mitochondria fused into aerobic prokaryote. That means fused into a cell, and it started living as a symbiont with the cell. With the evolution, it became a part of the cell, and it has its own genetic material, and it undergoes binary fission, and also the membrane that is present in the mitochondria resembles that of bacterial membrane. So all these evidences make up into one point that these mitochondria are bacterial endosymbionts of the cell. Thus, they are thought of as they had an independent life before. So now let's move into the basic structure of the mitochondria. The basic structure of, I can say, the shape of mitochondria is cylindrical, or it is sausage shape, but in yeast it is spherical, and it is also found in some other forms like filamentous, club shaped, racket shaped, or vesicular. The ultrastructure of mitochondria, if we talk about, it has two membranes, and the thickness of these membranes ranges from 6 to 7.5 nanometer, and both of these membranes are separated by a perimitochondrial space. This mitochondrial space is between the two membranes. The inner membrane, which has cardiolipins about 20%, and basically the membrane is, if you talk about its composition, it is made up of 40% lipid, 60% protein, and the inner membrane is selectively permeable, and it has about 80% of protein, and this 20% of the lipid that it contains is cardiolipids. And the tubular infolds that is present in the mitochondria inner membrane, these are called mitochondrial cristae, and these cristae plays a very crucial role in mitochondrial functioning, as these cristae have some numerous particles called as the oxysomes, or you can say F1 particles. These oxysomes have a base and a stalk and a headpiece that you can see over here with a magnified picture, and these cristae have having these oxysomes over here, as you can see in the magnified inner membrane infold. This is the inner membrane infold, and on the infold these are these oxysomes, and these oxysomes contain a very crucial enzyme called the ATP synthase, and this ATP synthetase undergoes oxidative phosphorylation and with the coupling of ATP. So this oxidative phosphorylation releases ATP. As we all know, mitochondria is called as the powerhouse of the cell. So because it provides the energy currency, which is adenosine triphosphate, short formed as ATP. So mitochondria is called as the powerhouse of the cell. The base and the inner membrane contains coenzymes of electron transport system. So this electron transport system is basically with the respiration, and we will talk about this in some other videos. So in this process also mitochondrial membrane takes part, and this mitochondria have their own DNA material, as I have told before. So this DNA material help in the production of their protein that is required in the processes that undergoes in the mitochondria. So this protein synthesis is basically carried out by the ribosomes that you can see over the matrix. So what is this? Where is this matrix? So after the inner membrane, there is a large mitochondrial cavity which is filled with dense gel fluid called the mitochondrial matrix. So this mitochondrial matrix contains high concentration of soluble proteins, nucleotides, some lipids, and the double membrane DNA. And a very crucial thing to know about these mitochondria is that they have 70 S ribosomes, whereas in a eukaryote you have 80 S ribosome. And this 70 S ribosome tells us another evidence that these are bacterial in origin, as bacteria also have 70 S ribosomes. So this cristae in the mitochondrial inner membrane plays very crucial role in the functioning. And also there is an enzyme called succinate dehydrogenase, which is the only enzyme of the Kreb cycle, which is associated with the inner membrane of the mitochondria. So this Kreb cycle is where you get many intermediates that is required for the cell functioning. This is another big cycle over here, process over here to understand. And you need to know that in the Kreb cycle, important enzyme, that is succinate dehydrogenase, is also produced in the inner membrane. Okay, so basically mitochondria is a very important organelle in the cell which undergoes some very crucial processes of the cell. Let's come to the functions of the mitochondria, like few other functions of the mitochondria. As I have talked earlier, mitochondria have their own DNA, though they are dependent upon nucleus and cytosol for some raw materials, and thus the structure and working of mitochondria is partially controlled by cell nucleus and cytosol. So they are not independent. They have a symbiotic relationship with the cell. And this mitochondria, as they are being controlled by cell nucleus and cytosol, so they are semi-autonomous. They are not independent. So all of us know that mitochondria is the powerhouse of the cell, and why it is so? Because it produces ATP, that is the energy currency of the cell, with the help of which the cell does many other cellular activities with the expense of this currency, that is ATP. And I have already told you that this ATP is released with the help of enzyme ATP synthetase, which is present in the inner membrane of the cell. Apart from this, the mitochondria matrix that is there, it has the site for cellular respiration. So the cellular respiration is the process in which the cell break down the sugar into energy. So the Krebs cycle occurs in the matrix, while the electron transport chain and the auxiliary phosphorylation occurs in the inner membrane of the mitochondria. So there is also an intermediate space that I have talked about, that is a perimitochondrial space, that has many types of enzymes which is required for many processes in the cell, and also in the matrix the pH that is there is very suitable for the Krebs cycle to undergo. And in the inner membrane there is a crucial enzyme, that is succinate dehydrogenase, which is produced, and this succinate dehydrogenase is important for the Krebs cycle, and the Krebs cycle is carried out in the matrix of the mitochondria. Apart from this, there is synthesis of many amino acids in the mitochondria. Also, elongation of fatty acids occur in the mitochondria. So, mitochondria basically is uh, the main function is giving energy to the cell. Apart from this, it is in semi-autonomous, and it has its own DNA. And one thing is that for the production of protein in the mitochondria, they have ribosomes. So the ribosomes in the mitochondria are a bit different. That is, their 70S, which is not usually present in eukaryotic cell. So from this is also an evidence which says that mitochondria is bacterial in nature. So they have 70S ribosome, whereas in eukaryotes they have 80S. So I hope this video will be useful for you. If you have any doubts, please do comment, and if you have liked my video, please do subscribe and like it. Thank you and bye bye till my next video. Bye bye.